Wonderful. Now we're recording, and I want to thank Erfan Vafaie, hopefully I said that correct, for being here to give the presentation today on Crape Myrtle Bark Scale. He is the Crape Myrtle Bark Scale guru for North Carolina, for North America, I guess, and he's going to help us learn more about the pest and how we can control it. So take it away.
Well, thank you so much. Yeah, thank you for having me and giving me the opportunity to talk about Crape Myrtle Bark Scale here. I want to mention that there are certainly a number of colleagues that I work with, and without them, we couldn't get this work done. Dr. Meng Gu with Texas A&M is the one that's kind of headed a lot of these projects and gotten a lot of funding to actually get a lot of this work done. So grateful for all those colleagues. And, just before we get in, just talking about crepe myrtles very briefly. They're a very popular landscape plant in the South because, very low maintenance, very beautiful flowering tree. And you can see there's many different colors of flowers. And it's been cultivated in the US for over 175 years, very common here in the South, valued at over 46 million in just the nursery crop sales, and that's back in 2009. And what's nice about it is very low, relatively low maintenance with very few key pests. So you kind of put it out there. They're very hard to kill. There is a popular pruning technique known as just topping, or sometimes referred to as crepe murder, right, where people just cut off the top. Not very encouraged from an aesthetic perspective, but what's kind of neat is, they always grow back. You can cut them back down to a stump, and oftentimes they can grow back. So they're very resilient, very low maintenance. And so some of these pests that we had in the past, right? So these are things that we've already had, include the flea beetle. All right, so this is a small little beetle that causes chewing damage, usually little holes in the leaves or chews on the edges of the leaves, but rarely will it defoliate the whole tree. Rarely is it really of any major concern. We also have a crepe myrtle aphid. They are usually found on the leaves of the crepe myrtles, and they're a sucking insect pest, very similar to what we're going to see, the Crape Myrtle Bark Scale, which is actually also present on this little twig. And they basically excrete this sticky solution known as honeydew that gets on the trees and branches, and then you get this concoction of molds that grows on that stickiness known as sooty mold. And we're going to see that a little later because we actually have Crape Myrtle Bark Scale doing the same thing. Get a little bit closer, you can see these aphids. They have these what looks like tiny little spines on them, but they're harmless to touch or to handle. But they can be, again, a pretty big nuisance on crepe myrtles. They're probably the second most important in terms of honeydew and sooty mold production on crepe myrtles in the landscape. You also have sharpshooters, such as the glass-winged sharpshooter that will feed on crepe myrtles. Again, not considered a major pest, although they will sometimes be seen on there, and they will shoot out honeydew. So if you actually observe one, or if you're sitting on a tree with several glass-winged sharpshooters, you'll feel the droplets falling on you, and that's that honeydew falling off them. So in 2004, my colleague, Dr. Mike Merchant, out of the Dallas, Texas A&M AgriLife Research and Extension Center, got a call from a landscaper about an issue where there are some white spots on these trees and sooty mold production on the bark, like nothing that I'd ever seen before. And so Dr. Merchant did a closer inspection and, took some nice photos and observations. And at this point, there had been no recorded bark scale on crepe myrtles, at least, especially in this type of abundance and felt scales. And so this was, the question was, where did this come from? Is it possible as a host shift? So not very common, but sometimes you can have insects that maybe they're specific to one type of plant, and in the, just the right environmental conditions or the lack of their host plant, they might adapt to and or start to establish on another plant. So in appearance, this scale is very similar to the azalea scale, known as Acanthococcus azaleae. And so the question at that time was this a host shift? Is this just a local population that has moved on to a new plant? However, another question was whether this was a new introduction. Was this a new invasive insect that had never been seen before in North America. And so there was this Acanthococcus lagerstroemiae that actually occurs in East and Southeast Asia on landscape crepe myrtles, which is actually where crepe myrtles come from. They're not native to the U.S. They are a cultivated plant here. And they did find this scale. And if you look close here, you can see this sooty mold on that bark. And that scale does occur there in the landscape. And so it's also possible at this point that maybe Crape Myrtle Bark Scale is a new introduction. And so they did some closer looks here, right? So on the left side, we have Acanthococcus azaleae, or the azalea bark scale. On the right side, we have the Crape Myrtle bark scale. And initially, it was actually noticed some slight physical differences, all right, that are actually distinguishable between the Crape Myrtle bark scale and the azalea bark scale. What they found was the scale occurring on crepe myrtles here was physically more similar to the Crape Myrtle bark scale, or the scale occurring on crepe myrtles in Southeast Asia and China, as compared to the azalea bark scale. And later, through DNA tests, it was confirmed that this was a new introduction. So it was an invasive insect pest. And invasive insects can be extremely problematic because they are coming from a habitat, into a new habitat where their natural predators are perhaps not there. Perhaps their host plant, such as crepe myrtles, are highly abundant, maybe not adapted to defend against them, so on and so forth. So they very quickly spread all across the U.S. You can see here from Texas all the way to Washington. And so they are kind of all over now at this point. And this is kind of what they look like. You can see these white spots on the tree, and they hurt the aesthetics of the tree. Now, it's very important that in this case, there were some, media outlets that, expressed the, an over-exaggerated view of how dangerous this scale is. So here's an example: invasive species killing crepe myrtles in green country. And it's not quite true. There's very little to no evidence that we've seen yet at this point that the Crape Myrtle bark scale will kill, especially a mature tree. If there are maybe new cuttings, they might overwhelm them. But really anything that's established in a pot or in the landscape is usually pretty resilient to the scale. And what it does, it just really hurts the aesthetics of it in the landscape. And if you're a nursery grower, it's a major economic concern because it basically means a stop sale or potential returned product. So if you have any of these white spots on there, that's a major economic loss. What's of another concern is the other potential alternate hosts based on the literature. So some of those include pomegranate, axlewood, Chinese hackberry, persimmon, common fig, soybean, border privet, and several rubus species. And so, there are some studies right now that we're doing to determine how many of these alternate hosts that are in the literature can our Crape Myrtle bark scale that's here in the US actually establish on. Here's a map showing the USDA hardness zone of where crepe myrtles can actually grow in the landscape. So it's anywhere in this green to brown zone here. So anything green and further south is where we can have crepe myrtles actually found in the landscape, with the southern states having abundance of them. So they are very popular in the landscape. We have already found Crape Myrtle bark scale on Beautyberry, on Calicarpo species in the landscape as well. So here it is established on it. And so again, that's another potential alternate host that's now been confirmed, not only in controlled studies, but actually naturally occurring in the landscape. So something to watch out for. You can see again those white spots, and you can see a whole lot of production of sooty mold on the bark and on those leaves. You see all that black on there. There's 5 plant species confirmed to be hosts, furthermore in actual greenhouse studies. So we got this American Beautyberry and pomegranate has been confirmed that they are potential suitable hosts for reproduction of Crape Myrtle bark scale in a no choice kind of a cage study. So it's perhaps just a matter of time before the populations outside really establish on these. We also have a few other, so henna, hymia, and winged loose strife, all of which have been confirmed again in lab studies, and several other loose strifes, 3 loose strife species that have also been confirmed as potential hosts in the landscape as well. So this is that sooty mold. You see that black mold growing on that bark. Those large white spots we typically see are either female egg sacs or male pupa. These small things here are the immatures. Very hard to see with the naked eye. You usually require some level of magnification to see them. And so, you can have a population of Crape Myrtle Bark Scale on that branch without really noticing it yet. It's not until a little bit later that you might start seeing female egg sacs or male pupa. We'll see here in just a moment what I mean when I refer to those. But you can see these immatures are very small, right? This is the tip of a needle right here, so they're very small. When they first emerge, they have legs to crawl around, find a place to establish. From there, they are typically quite sessile, so they're not moving around a whole lot after that. And they're feeding on the sap through that bark. And from that sap, they're trying to get nitrogen. With that nitrogen, they can make more amino acids. With amino acids, make more protein. With protein, make more babies. So these are just baby-making machines. As they're filtering through that sap, they're going through a lot of this solution, a lot of the sugary solution. So lots extra, which they excrete again. So they're pooping out these droplets of sugary solution known as honeydew. And that's what, when it creates a layer on that bark, this concoction of molds grows on. You get that sooty mold. And the sooty mold itself will not kill the tree. It'll just really hurt the aesthetics. If it gets on plants or on the leaf surfaces, it can potentially reduce the photosynthetic ability of that leaf. So the ability for it to use the energy from the sun to grow essentially. And this is an egg sac right here, opened up. So again, those eggs are very heavily protected under this like waxy layer, right? So when we're talking insecticide applications, they're not going to work very well against these eggs because they are quite protected inside that waxy layer. So here's just, a try image real quick of, a highly infested branch here on the far left. The middle is a female egg sac and a male pupa. So the female egg sac, the female basically, as she starts to mature, so these are the immatures on the far right, as they start to mature, they start to form this waxy layer on the outside. The female lays all her eggs inside there and she shrivels up and dies. And then all those eggs, they come out as new crawlers and they'll find a new place to establish. The males, on the other hand, will metamorphose. So as they're becoming more mature, they go inside this little pupa and then they become a winged male. So just like a caterpillar to a butterfly, they are transforming to a winged male and then they can disperse in that way. You can see they're quite distinct, right? The female egg sac is quite round and large, whereas the male pupa is quite oblong. It's not quite as round, it's more longer, but also smaller. So this is an example of a Crape Myrtle Bark Scale nymph, a little bit closer up. And this is a female taken outside of her egg sac, so just before she lays those eggs. And if you do a dissection of these females, so here I dissected about 20 females and found anywhere between 60 to 250 eggs per female, right? So it might even be on the higher end of that. Maybe they're still developing some of those eggs when they're in that egg sac and continue producing them. But you can see there's a good number of eggs produced per female. And this is what the winged male looks like. Again, very small, hard to see with the naked eye, but it's the males that can fly around and disperse. It's thought that the females disperse perhaps by wind or jumping onto larger animals like birds or larger insects. It's been shown in other scale insects that that's some of the ways they can disperse, or by movement of plant material. So we are moving around Crape myrtle cuttings, in the back of pickup trucks and whatnot, and it can disperse very quickly that way as well. Their current distribution, again, is kind of relatively spread. Now, this is reported by people, right? So it's possible that it's in these other areas as well, but just has not yet been reported. You can contribute to this report map. So if you do not see your particular county or region represented, and you know that Crape Myrtle Bark Scale is there or want to contribute to this, you can go to edmaps, that's eddmaps.org/CMVS. And you'll get a report form that looks something like this. I'll show later, you can also go to our stopcmbs.com website. And here you can actually report it. You can do it from your phone and use location services to track your GPS, and you can enter it in there. And then myself or a few other colleagues can go in and confirm that. Make sure you take a nice photo. It's a lot easier that way to verify if you have Crape Myrtle Bark Scale or not. So today I want to cover, when we're actually talking about some of the studies we've done so far, Crape Myrtle Bark Scale, trying to understand them and how to manage them. I'm going to cover 4 main objectives out of several objectives that our larger national team is kind of covering. One's going to be on, are there any cultivars that are resistant? Looking at population dynamics. So that's kind of when do they first start coming out of those eggs, and do we have an increase and decrease in populations? Looking at population control using insecticides. And lastly, the role of beneficial insects, or oftenly referred to as natural enemies, predators that are occurring in the landscape. So we're talking cultivar susceptibility. We did this quick little observational trial just north of Dallas. So this is where we actually first saw the Crape Myrtle Bark Scale, where it was first observed, I should say. It also happens to be known as the Crape Myrtle capital of the US in a sense. So there are over 120 varieties of Crape Myrtles in the landscape there. They're all different types and have a history of infestation. We went through and collected at least 3 branches per tree, at least 5 trees of a given cultivar, and looked at those under the microscope and counted all the scale. Now, before I go on to the next slide, this is a major disclaimer, right? So the actual quantities that we see here are not going to be super reliable as an indicator of how susceptible different cultivars are. And the reason here is this is purely observational. So if Crape Myrtle Bark Scale started at a certain site, or someone had moved some infested materials there, you're going to see some higher numbers of Crape Myrtle Bark Scale there, not because those cultivars are particularly more susceptible, but perhaps just because of the way the scale was moved around. So you can see here on my x-axis on the bottom, I have several different cultivars that we looked at, from Tuscarora all the way to Natchez. And on the y-axis, I have the average number of scale that we counted per centimeter of branch. So you can see here, if you were to take this as factual of what is susceptible and what isn't, you would think that Tuscarora, Lipan, and pink ruffles are really bad and you'd want to avoid those, whereas, Miami, Sarah's Favorite, Twilight, and Natchez appear to be very resistant, and maybe those ones are better. But if you live in Smith County, which is where I'm based out of, most of our Crape Myrtles are Natchez, and those numbers are very high. So really the takeaway from this is that from all the cultivars that we looked at, there were none that had 0. There were none that were fully resistant. And now there are some controlled studies that some colleagues are doing to determine if you put 10 scale per tree on each of the different cultivars, are there any differences in the rates of population growth? And that's one way, in a controlled way, to know whether certain trees have a little bit more defenses or resistance against the scale versus others. This was another thing, we had some people that would say, well, maybe we should call this the green Crape Myrtle Bark Scale because I haven't seen it on the purple leaf. We have seen it on the purple leaf Crape Myrtle Bark Scale, Crape Myrtles as well. So even these new purple leaf ones, unfortunately, are not resistant to the Crape Myrtle Bark Scale. So our second objective was looking at population dynamics, right? So if we can know when Crape Myrtle Bark Scale first starts coming out of their eggs or first starts moving early in the season, we can know perhaps when's the best time to manage our insecticide applications or management strategies to quickly knock them out before they become problematic. So how we did that was using this highly sophisticated method known as double-sided sticky tape you get from an office or Home Depot. And we're using this permanent double-sided sticky tape and wrapping it around the branches. And we do at least 5 branches per tree and at least 3 trees per location. So you'll see we monitored scale throughout Texas, a few different places in Texas, Louisiana, and Arkansas. And we would collect these tapes on a weekly basis and replace them and count all the scale on those sticky tapes. And the idea here is when those crawlers right? Then when they first come out of the eggs, they are moving around quite a bit on that tree and they get stuck on that double-sided sticky tape. So by looking at those numbers week after week, we get an idea of the relative change or relative abundance of the scale over time. Here's what it looks like under the microscope when we take that double-sided sticky tape, put it on graph paper, and you can see these are all immature crawlers here that have gotten stuck on that tape. And so we can count those again and standardize it to number of crawlers per square centimeter per week and tabulate that. So these are all the different locations in which we monitored. So Dallas, Tyler, Huntsville, College Station, Shreveport, and Houma. This data was actually just published today through the Journal of Environmental Horticulture. And it is open access, so you can actually go and look at the specifics of how we conducted this work and the results of the data. So if we look at 3 separate years, all right, so follow with me here. Each of these boxes represents one of the different locations. On the x-axis here, we have the month of the year, so January to December. On the y-axis, we have the average number of Crape Myrtle Bark Scale. This is in the year 2015, all right? So you'll notice that for 2015, all of our scale populations start to increase towards a peak, right? So right here, right here, right here, right about here, they start to increase towards a peak by around mid-March to beginning of May. There are some sites that started monitoring a little bit late, so I'm not including those in terms of when those peaks first occurred. If we look at 2016, we see it's a much tighter window, but again, is kind of between this mid-April to beginning of May, when all of our locations start going towards an initial peak in crawler numbers. Lastly, in 2017, you'll see again, this case about mid-March to beginning of April. So there's a bit of variation, but it's kind of between that mid-March to mid-April is when we start to see an increase towards a peak for all locations, right here, mid-March to about beginning of May. And we tried using climate data as a way to predict Crape Myrtle Bark Scale emergence, and we found at least preliminary, this data that we analyzed most recently was no better. Climate was no better at predicting initial emergence. It's actually worse at predicting emergence than just calendar date. So for now, at least for our region, what we suggest is if you're trying to knock out that population, and this is for growers, right? Landscapers, you're trying to manage them below a certain threshold, but for growers, it's vital for them to knock out that population, that they need to be doing some contact spray applications right around this mid-March to beginning of May time period. So as a part of this as well, there were some observations, right, that people said, well, what happens if, you're putting your tapes higher, so up on these upper branches, or lower? Because some people, had made this anecdotal observation that it looks like the scales move up to the upper branches throughout the season. So they're slowly migrating up to the upper branches. This is also relevant because, here, you can see here, so these are lower branches and upper branches. This is also quite relevant because, in our methods, the way we were placing our double-sided sticky tape to standardize it across locations was to place the tape at breast height. The problem is breast height can be very different when you have a height differential, right? So I'm 6'7". Right, my colleague here, I think she's about 5 foot, Dr. Meng Meng Gu, who's kind of headed a lot of this work. And you can see this is the lower branch, right? This is the upper branch right here, right? So we're trying to determine, are there any differences? And they looked at this on 12 different trees, all in College Station. Now this next slide, I warn you, is not going to be very pleasant to look at, but just focus on, how I'm going to walk you through this here. Each of these boxes represents a tree, all right? Tree 5, tree 6, tree 7, 8, all the way to tree 12. On the x-axis for all of them is the month. On the y-axis for all of them is the number of scale. The different colors represent upper or lower branches. The takeaway here is that upper and lower branches, the red and the black lines for all these boxes, more or less trace each other. They have slightly different amplitudes in some cases, right? So some might be a little bit higher than others, but in terms of when they start increasing and decreasing is pretty consistent. So when you're looking at relative abundance or relative change of scale over time, it seems, at least based on this data, it doesn't really matter if you're putting your tape on lower branches or on upper branches. All right, so now we're getting into management. And we've done a number of trials. This one in particular was in the landscape. This one particular church that had about 75 crepe myrtles in their landscape. Oh, excuse me. And they had a history of infestation. So we tried a few different treatments here, which we'll kind of go through. And each treatment was replicated 7 times. You can see here my summer employee at the time, he's now a PhD, Dr. Patrick Ridzak, is wrapping these branches with double-sided sticky tape. So that was, again, our method of determining how well these insecticides worked. We applied our contact insecticides twice on a 2-week interval. So one of them was a horticultural oil and insect growth regulator, azodiractin. Another one was bifenthrin, a pyrethroid. The last one was acephate. We also applied just the City of Tyler water to see if our water is effective at managing the scale. All right, in our drenches, we actually mixed up in buckets and poured around the base of the tree. We only applied it once. We tried both imidacloprid and dietephyran. If you're a homeowner, imidacloprid is readily available at your Home Depots and Lowe's. If you're a grower or landscaper, you're going to have access to dietephyran and some of those other insecticides I mentioned earlier as well. When it's on a slope, right, you got some mulch, we're usually digging a small trench around the base of that tree before we do the drench to make sure that water actually goes down to the root zone of the crepe myrtles. So here's what the data looks like. All right, so on the x-axis here, okay, I have date, all right, and on the y-axis, average number of scale. This is when we did our spray, our insecticide spray applications. Now follow with me here, just the water application. So the reason why I'm showing you this data, and it's very important to have the water in there as well, is because it's very easy for, and I've found a lot of information online that says, this will work or that will work. And without the data to support it, right, without the ability to look at data and just understand the basics of it, it's hard to discern what is true and what is not. So in this case, you'll notice our water-only treatment, so here's our scale numbers for water, this blue, starts to increase towards a peak, which is what we've seen earlier, right? So we get this first peak and it kind of goes down after, right? So then it goes down, and then we get the second peak later in the season, right here in June, right? And then it comes back down around August. So if we looked only sorry, that was July. If we looked only in June here, where the numbers are very low, and didn't have our water treatment, it would look like all of our insecticides worked very well, right? So here we're comparing everything to our water treatment. And especially when those water treatments are high, that scale population is high, is a good time to see, well, how do the insecticides do compared to it? So if we look at just acephate, which is this red one here, you'll notice it increases perhaps even higher than the water. It's not significantly different, but it does increase at the same rate as the water decreases and increases again. And you'll see a little bit later that there's actually data from a colleague, again, Dr. Mike Merchant, that demonstrates that acephate can actually kill all the beneficials and does nothing to the scale. So you could potentially be promoting the scale population by using acephate. If we look at Suff oil and Molt X, that's the green here, you'll notice we get some suppression compared to the water, but really not sufficient to give us really good control. So maybe in a landscape type setting, maybe that's okay, but when we're talking in a nursery, this is going to be considered insufficient. If we look at Talstar, that's the orange, you'll notice that it just starts to increase here, and then it goes right down, and it stays down for a good while until that second peak. So Talstar or bifenthrin gave us very good short-term control. If we look at our Merit and Safari drenches, that's these two purple colors, you'll notice that first peak go up really high, comes down, and by about early June, those two purple colors disappear off our graph. So these two give you very good long-term control. However, they do not work right away. So we applied them here near the beginning of April, but it takes about 60 days for them to become active. And that's right about this mark here, right? Because they're taken up by the tree. It takes a little while for them to be taken up before the insects actually consume it. So if you're doing a drench application, we'll see a little bit later on, it's more beneficial to apply it quite a bit earlier, as soon as those leaves start budding out. So for us, that would have been just a few weeks ago or as soon as possible. I have in this presentation, some details about applications and application timing. I'll show you all later where you can actually download this presentation, or you can obviously view this recording. But if you actually just want to download the presentation, take the tables out of it and whatnot, you're more than welcome. This is all published data as well and open access, so anyone can get to it. So in this next trial we did, we looked at potted or containerized crepe myrtles. So this is more of a nursery type setting. Again, we used double-sided sticky tape and those 5-gallon pots. We tested a number of different products. And again, I'm going to kind of walk you through those. So this is kind of what our trial looked like. You can see here, over here is things are not quite ready yet. When they're not trellised up, they'll just kind of topple over. But we provided enough space between them so the crawlers are not going from one tree to the next, and randomized our treatment applications. So here's all of the data together, all right? And I'm gonna kind of break it down for you piece by piece. So here on the x-axis, again, we have date. We also have weeks after treatment, so 0, 4, 7, and 19. And I've kind of selected these dates to show you because they're kind of the most important in terms of when you can see those scale populations. On the y-axis, again, is the average number of crawlers. So you can see mid-March kind of low, by the end of April, quite high, and then we have that decrease again. Now, if we look at what I call now our NUCIT option, all right, so that's a combination of a dinetefuran drench or safari drench and bifenthrin spray application. So this is like really hitting them hard. We can see again, so this is our pre-application numbers, and then afterwards, week 4, very low, and from there it stays quite low. So this option will with reasonable confidence, give you very good control. If we look at some of the others, so just a dinetefuran drench, so if we're not doing the bifenthrin spray, we still get very good control, although initially, there's a bit more variability. You might have slightly higher numbers, but then it catches up and remains the same. We can also do a dinetefuran spray. So if you're in a nursery setting and drenching every pot is not practical, the label does have a bark spray on there. And you will get some of that insecticide moving in through the bark as well. So it'll kind of work a little bit as a contact and then also a little bit as a long-term drench as well. So that's also a good option. Imidacloprid, you'll notice in this case, this drench did not work that well. And this is the only thing, there's been twice. So myself, this is my one experience, and Dr. Mike Merchant in Dallas, he has some data where the imidacloprid drench did not work in one case as well. We cannot quite explain why. So at times, imidacloprid may not work all that well for whatever reason. All right, so then we got pyriproxyfen is an insect growth regulator. You can see this purple come up and then goes right down. Gives you very good contact control. We have, we have cyantraniliprole and flupyridiferone. These are 2 relatively new insecticides that are right over here. Can give some good control a little bit later in the season. And lastly, we have buprofizine, which is another insect growth regulator. And you can see here that it works quite well too. And I apologize, I think earlier I referred to this one as pyriproxyfen, and it's not actually that one, it's this one right here. So pyriproxyfen actually works very well as a contact insecticide. So these are all contacts. Then we have several others that did not work well. And again, so if I did not just discuss it, it's because they did not work well to manage the scale out of this list. Again, I have all those spray application details and rates that we used in there that y'all can take a look at and use. And that data kind of summarized, if we look at just near the end of April, again, we can see Safari Drench. It's a bark spray mixed with some capsule, Fulcrum, which is our pyriproxyfen, And then we also had good control from Talus, Talstar, and the Safari Drench as our nuke options. All of them gave us early and some reasonable long-term control as well. Now I always get the question, right? So here's a case, right? So very well treated, our nuke option is very nice and clean versus our untreated check. You can see all that city mold and all the scale on there. Now I often get the question, whether it be landscapers, nursery growers, can I clean off that city mold? What can I do to take that off? And so we tried a few different things, from brushing to high pressure water spray to hydrogen peroxide, and a few different things. So here's, again, Dr. Patrick Ridzak kind of brushing off that city mold. You can tell from his face, he's not having a whole lot of fun here. So it is a labor intensive job. In the nursery setting, it might make more sense to try and bump up your crepe myrtles to a higher, larger pot size, wait for them to shed their bark, and then keep them clean from there. Whereas in a landscape setting, if you're a homeowner, have at it. There hasn't been a whole lot of data to support that you can control the scale this way. There hasn't been a whole lot of data to show that this will, relieve some of the aesthetic problems in the long term. But if it makes you feel good, and it does, at least for the short term, make it look a bit better, it doesn't, to our knowledge, doesn't do any harm to the tree. So that's okay. Now we do have a number of beneficial insects already, naturally occurring beneficials that are feeding on the scale here. So if you ever see these things crawling up and down the tree, these are lady beetle larvae. This is most likely Harmonia axaritis, also known as the multicolor Asian lady beetle. And it usually feeds on aphids, the Crapemyrtle aphid, usually not on a scale. Now here on the left, you'll see this thing. It looks a lot like a mealybug, or looks a lot like another scale, but this is actually another native lady beetle species in its larva, in this larval form. So before it's mature, it'll feed on scales. So we've observed this in a lot of situations. So here's an example of two different lady beetle species. And here's an example of Hyperaspis larva, the larva of this one right here, possibly. And so you can see that as an immature, they look quite different, and they have this waxy outer coating. As adults, they're a little bit different than what we're used to when we think about ladybugs or lady beetles. In this case, they're black with red dots rather than red with black dots. And if you ever see this on a branch, right, I often get photos of these, people concerned that these are bad or what to do to control them. This is actually when the larva is pupating, the larva is becoming mature, they'll form this pupa, this cocoon type thing, and they often do it in aggregations together. I suspect, and maybe in the literature there's something about it, but I suspect it's some kind of defense against predators by kind of being together, and maybe they reduce the chance of them being all eaten, perhaps. But these, after they come out, those exuvia, we call it, right? It's like the shed skin of the pupa stays on that tree, and it can stay on there for over a year. So I had actually taken this photo, and then a year later, I was walking past the same tree. I observed the same thing, and I thought, oh, wow, that's really cool. I should take a picture. And right after I took a picture, I realized it was the exact same ones I'd taken a photo of the year before, right? So they can stay on there pretty long, not bad, right? So these are lady beetle exuvia. They're considered beneficial. And here it is from a colleague, Mike Gilder, who's a master's student at Texas A&M in the same lab that I also participate in. And he's done these nice illustrations. He's working on cataloging all the different naturally occurring beneficial insects that feed on the Crape Myrtle Bark Scale. So that again includes a number of lady beetles and as well as green lacewing larvae that will also feed on the scale too. And here is a nice little table that he's starting to form, and he's starting to look at food web interactions and what things in the habitat may be contributing to more beneficial insects. So in that way, we can look at maybe habitat or landscape manipulation to promote these beneficials and to manage their scale. And these are very important, right? So we can't underestimate how important these naturally occurring beneficials are. We have, again, Dr. Merchant did this little trial here where he wanted to know what happens when you take out the beneficials. So he did either, imidacloprid drench or no drench, right? So he's like treating for the scale. Then he also had a lady beetle treatment. So he's like trying to kill the lady beetles and not killing the lady beetles. What we see here, all right, on the x-axis, in this case, there's nothing because the different colors represent the different treatments. And on the y-axis, we have number of Crape Myrtle Bark Scale. So here, these four bars right here is what happens when you treat for the lady beetles only. So we're just killing our beneficial beetles. Whereas if we don't treat at all, if we don't apply any insecticides, so you'll notice if you're applying something that kills only the beneficials, we get about a 3- or 4-fold increase in the number of scale. And some of those treatments he used to kill just the lady beetles include carbaryl or 7-dust. And I know that's very common in home use or by homeowners. So be cautious. This 7-dust is not something that works against the scale, and if anything can exacerbate the problem. You'll see here, this is what happens if you treat for the beetles and the scales. So if this is still too high for you, too high of a number, you can do a treatment of imidacloprid and knock those scales down furthermore. And you'll notice this one bar here, this is imidacloprid, right? So I mentioned sometimes for whatever reason, sometimes does not work. So that's something just to keep an eye out for. We also have a graduate student, Kenneth Maslowski, that's working on what's known as importation or classical biological control. So he's actually looking at samples from back in China to see if there are any beneficial insects that we could import and would have very little to no, should really have no negative impact on our other beneficial insects. And so he's actually found 4 groups of parasitic wasps. So these are wasps that lay eggs in or on the scale, and will basically, kill that scale and produce more wasps. So he's rearing them in quarantine, determining the efficacy against Crape Myrtle Bark Scale and determining kind of some of those non-target impacts. So to summarize what we spoke about, right? So when we're talking about population dynamics, the crawler populations appear to be most active around, it was like beginning of May is when we have that peak. We want to hit them before that peak. So anywhere between mid-March to near the end of April is when we start to see that increase towards a peak. In terms of natural predators, we have mostly lady beetles that are feeding on them, and they can provide about 75% suppression in the landscape. And in terms of management, we're finding bifenthrin, buprofusine, and pyriproxyfen to be the most effective as contact insecticides, imidacloprid and dinotefuran, with dinotefuran being a little bit more reliable for our drench applications. And I do have, so again, you can extract this table, a nice little summary table if you need to look at what things so far from what we've tested kind of work well. So here I have great, good, moderate to good, poor, or inconclusive, and we're conducting further trials to determine whether they work. I do encourage you to check out our stopcmbs.com website. And here, let me go real quickly into it right here. So it's been recently updated to provide resources information for you. You can, if you go to our updates, you'll see kind of recent updates and information from other graduate students or other researchers related to Crape Myrtle Bark Scale. So for example, containerized Crape Myrtle Bark Scale trial here, you will find this table that has information about summarizing all the insecticides that I've tested so far and what has worked, whether it's worked well or not. This particular presentation, if you go under present resources and presentations, you'll find both the PowerPoint and the PDF for this presentation here. So you can download it and, extract pieces from it, present it, do whatever you so wish from there. And that concludes the presentation. So I want to thank you kindly for your time.